The messenger RNA has left the nucleus and is now in the cytoplasm. Here, the message will be translated into a protein sequence. Here's the messenger RNA in yellow. It is held in place by the ribosome. The ribosome appears in lavender, and this is the site for protein synthesis. The ribosome has the ability to read the message within the messenger RNA. Ribosomes can be free in the cytosol or they can be bound to the rough endoplasmic reticulum. So protein synthesis occurs in both these locations. The ribosome reads the code. The code is carried by the mRNA. So what's the code? The mRNA, the language of the mRNA, has many words, and each word has only 3 letters. These 3 letters are really the nucleotides. So the nucleotide sequence is read in triplets because each word has only 3 letters, and each triplet code is called a codon. And each codon represents an amino acid. As we know, there are 20 different amino acids, but we actually have 64 codons, which means a single amino acid can be represented by more than one codon. This might seem a little confusing, but there is a dictionary that can help us. And that dictionary has both amino acids and the codons. It's a codon amino acid dictionary. So here is the codon dictionary. The small letters that you see here, PHE, LEU, another LEU, ILE, all of these are the amino acids. PHE is phenylalanine, LEU is leucine, ILE is isoleucine, and so on. Codon and see what the amino acid might be. So let's pick CAA. CAA goes with glutamine. So there's glutamine. Let's say that the next codon that we have is UAC. Here's UAC, and the amino acid that goes with it is TYR, that is tyrosine. Let's try another codon, AUG. Here's AUG, and it matches with MET, which is methionine. This is usually the first codon that the ribosome encounters, and we call it the start codon. Let's look for another codon, UGA. UGA stands for the stop codon. If you have a start codon, there is a stop codon, which means stop reading the message. We are done with making the protein. The first step in translation begins with the ribosome holding the mRNA in place. There's the ribosome, and here's the mRNA with its codons. There is yet another type of RNA, and we call it the transfer RNA or tRNA. tRNA has 2 ends. At one end, it picks up an amino acid, and at the other end, it has an anticodon, which is complementary to the codon in the mRNA. There are several different types of tRNA, and each tRNA is highly specific to an amino acid. The first step is for the tRNA to pick up its amino acid. To do this, it needs energy, and that energy is provided by one ATP molecule. The process of bringing the first amino acid to the site of protein synthesis is called initiation. Here we have the ribosome holding the mRNA in place. There is enough room to hold 2 codons inside each ribosome. The first codon is AUG. And the tRNA that comes in should have an anticodon, which is complementary to AUG. So it would have to be UAC. It brings an amino acid with it, which is specified by the AUG, which should be methionine. The second tRNA has its anticodon that matches with CCA, and it brings the second amino acid. The second stage of translation is called elongation. We just saw the first tRNA bringing with it its amino acid, which is methionine. This first tRNA hands off the methionine to the second tRNA. Once it does that, the first tRNA is ready to leave. It leaves the ribosome and goes off to find its amino acid. Now the second tRNA shifts its position because the mRNA shifts down one place. So here's the second tRNA holding on to two amino acids. There is an enzyme that helps this transfer of amino acid from one tRNA to the next, and this enzyme is called amino-acyl transferase. This process is repeated when a third tRNA arrives with its amino acid. Then the second one will hand off two amino acids to the third tRNA. So the polypeptide chain continues to elongate as amino acids are added one at a time. So here we have a tRNA with a very long polypeptide chain. Here is the amino acids. The amino acids are held together by covalent bonds known as peptide bonds. There is the previous tRNA that has broken free from the ribosome. Finally, when the ribosome encounters one of the stop codons, such as UGA, the entire process comes to a grinding halt. It's like brakes have been applied, and the last tRNA releases the polypeptide chain. Once it releases the polypeptide chain, the tRNA is free to leave the ribosome. Now the ribosome has finished its job, and so once the tRNA leaves, the ribosome also separates from the mRNA, and the entire process can be repeated. So this stage is called termination. The entire process of protein synthesis, starting from transcription in the nucleus all the way to termination, only takes about 60 seconds.